Honey never spoils. Bury it, forget it, dig it up 3,000 years later. It's still edible. And it still kills the bacteria that would kill you. Egyptian surgeons packed wounds with it in 3000 BCE. They didn't know why. It just worked. For 99.9% of human history, there was no such thing as an antibiotic. No penicillin, no hand sanitizer, no sterile gauze, no hospital with locked doors and clean floors. A scraped knee could kill you. Childbirth could kill you. A splinter, infected and ignored, could kill you. And yet humans didn't go extinct. They built empires. They crossed oceans. They raised children who raised us. Somehow, without knowing what a bacterium even was, they were already fighting one. So how did they do it? Not with luck, not with prayer alone. With a toolkit, built by trial, death, and repetition. Honey that never spoiled. Bread deliberately left to rot. And maggots placed on open wounds on purpose. 3 separate solutions. 3 separate mechanisms. None of them understood at the time. All of them, it turns out, were correct. Start with the jar. Archaeologists digging into tombs near modern-day Luxor, Egypt, found sealed clay pots. Inside, honey. Not fossilized, not crystallized into something unrecognizable. Honey. Soft, spreadable, edible honey. 3,000 years old. Here's what's fascinating about that. Honey doesn't spoil because bacteria can't survive inside it. Not struggle to survive. Can't. The mechanism is almost embarrassingly simple. Honey is roughly 80% sugar and less than 18% water. Any bacterial cell that lands inside it immediately starts losing water through its own membrane, pulled outward by the sugar concentration around it. The same process that makes salt kill slugs in a garden. The cell shrivels. It dies of thirst, essentially, in a pool of food. But that's only the first weapon. Bees add a second one on their way into the hive. An enzyme called glucose oxidase, secreted from a gland near the bee's mouth, breaks down the sugar in nectar and produces small, steady amounts of hydrogen peroxide. The same chemical used to disinfect cuts today. Not enough to burn tissue, just enough to kill what's trying to grow in it. So honey isn't one defense. It's 2, layered on top of each other, built by an insect that has no idea what a bacterium is. And ancient humans didn't know the mechanism either. They just noticed the pattern. Wounds packed with honey healed. Wounds left bare, more often, didn't. The Ebers Papyrus, an Egyptian medical school dated to around 1550 BCE, lists honey, animal fat, and plant fiber as a standard wound dressing. Not a folk remedy on the margins, a documented, repeated medical protocol.
Sumerian clay tablets from centuries earlier described the same idea. Honey, applied directly to stop a wound from turning. Even Hippocrates, writing in Greece around 400 BCE, prescribed honey mixed with vinegar for open sores. Different language, different continent, same conclusion. Researchers at Cardiff Metropolitan University, led by microbiologist Rose Cooper, spent years testing that ancient formula against modern bacteria. In lab conditions, Manuka honey from New Zealand inhibited the growth of Staphylococcus aureus, including strains already resistant to conventional antibiotics. Bacteria that had evolved past our modern drugs still couldn't survive something bees have been making for 100 million years. Here's the part nobody talks about. Manuka honey has a third weapon the Egyptians never even knew existed. A compound called methylglyoxal, found almost nowhere else in nature at these concentrations, derived from a sugar in the nectar of the Manuka flowering shrub, native to New Zealand and Southeast Australia. It doesn't evaporate. It doesn't break down when diluted. It doesn't lose potency sitting in a jar for years. Which is part of why the honey found in that Egyptian tomb was still, technically, sterile. Not preserved. Actively hostile to anything trying to grow inside it 3,000 years after the person who sealed the jar died. Today, hospitals use medical-grade honey dressings, sold under names like Medi-Honey, specifically for wounds that won't respond to antibiotics anymore. Diabetic ulcers, post-surgical infections, the cases doctors call last resort. The World Health Organization now lists antibiotic resistance as one of the top global public health threats facing modern medicine. And one of the substances hospitals are reaching back for is functionally the same jar the Egyptians were packing into open wounds 5,000 years before germ theory existed. Not a coincidence. Not a lucky guess that happened to work. A pattern, observed and repeated by people with no microscope, no theory, no idea what a cell was. Just a wound that got better and a wound that didn't. That's the first tool in the ancient kit. Not medicine in the way we define it now. Chemistry, discovered by accident and kept because it worked.
But honey only does one job. It stops things from growing. It doesn't clean out what's already dead. And dead tissue sitting inside a wound is its own kind of danger, a breeding ground the honey alone can't reach. Ancient healers noticed that too, and the solution they landed on is the part of this story most people can't watch without flinching. Not because it didn't work, because of what it actually was. So what do you do with a wound honey can't fully clean? For most of history, doctors had two options. Cut the dead tissue out, with a blade, without anesthesia, and often without surviving the blood loss. Or let it sit and watch the infection spread until amputation became the only option left. There was, unintentionally, a third.
Battlefield surgeons across centuries kept noticing something they didn't ask for. Wounds left too long in the field, in warm weather, attracted flies. Flies laid eggs. Eggs hatched into larvae. And soldiers whose wounds became infested by accident, by neglect, by circumstance, sometimes healed faster and cleaner than soldiers whose wounds stayed sterile. Not despite the maggots, because of them. Napoleon's chief military surgeon, Dominique Jean Leray, documented this directly during campaigns in the early 1800s, noting that maggot-infested wounds showed less putrefaction than wounds kept clean. He didn't understand why. He just wrote it down. Confederate and Union surgeons noted the same thing independently, decades later during the American Civil War. Different war, different continent, same observation. Here's the mechanism, confirmed roughly a century after Leray. Certain fly larvae, specifically the green bottle fly, Lucilia sericata, only eat dead and dying tissue, not living flesh. Their digestive enzymes dissolve necrotic material on contact, while leaving healthy tissue almost completely untouched, a precision no scalpel of the era could match. And as they feed, they secrete compounds with their own antibacterial properties, effectively sterilizing the wound bed as they clean it. Two jobs, one organism, no training required. In 1929, an American orthopedic surgeon named Dr. William Bayer formalized this into an actual medical treatment at Johns Hopkins Hospital in Baltimore. He'd first seen it during the First World War, treating soldiers whose wounds had gone untouched for days in the field. He deliberately introduced sterilized maggots into the wounds of patients with chronic bone infections, cases that had failed every other available treatment. Of the first 21 patients treated this way, Bayer reported that all of them were covered, debrided cleanly, without a single case of the infection spreading further. Before penicillin, before sulfa drugs, before any pharmaceutical treatment for infection existed at all. It's called maggot debridement therapy. And here's the part nobody talks about. It never actually went away. It nearly did once antibiotics arrived in the 1940s. Doctors dropped it almost overnight. Faster, cleaner, no larvae involved. But by the 1980s and 90s, antibiotic-resistant bacteria started appearing in hospitals faster than new antibiotics could be developed. And maggot therapy quietly came back. Physician Ronald Sherman, working out of the University of California, Irvine, spent much of the 1990s re-establishing it as a modern clinical treatment, publishing case after case of wounds that antibiotics alone couldn't close. The U.S. Food and Drug Administration cleared medical-grade maggots as an approved treatment device in 2004 for diabetic ulcers, pressure sores, wounds that antibiotics could no longer touch. Same organism, same mechanism Leray watched happen by accident on a battlefield 200 years earlier. We just needed to run out of alternatives to remember it had never stopped working.
So that's the second tool. Not a substance this time, a living organism, recruited without being domesticated, doing a job with a precision our own hands couldn't replicate. Honey stopped bacteria from growing. Maggots removed what honey couldn't reach. But neither of them could do what the third discovery could do: kill an infection that had already taken hold, deep inside the body, where nothing could be packed or applied directly. For that, humans needed something stranger, something that, by every instinct, looks like exactly the wrong thing to eat. So what did they do when the infection wasn't in a wound, when it was a fever, a cough, an illness with no visible edge to clean? Across dozens of unconnected cultures, the same strange remedy shows up in the historical record. Ancient Egyptian physicians prescribed moldy bread applied directly to infections. Serbian and Chinese folk medicine, centuries apart and with no contact between them, independently used moldy soybean curd and moldy bread on wounds and sores. In parts of the medieval world, cobwebs mixed with moldy bread were packed into cuts on purpose. Not a superstition passed down blindly,
A pattern discovered independently over and over by people separated by oceans and thousands of years. Here's the question worth sitting with. What were they actually doing? Bread left in a warm, damp place grows a specific kind of mold, most commonly a species in the genus Penicillium. Yes, that Penicillium. The same genus Alexander Fleming would famously discover in 1928 when a stray spore contaminated a petri dish he'd accidentally left uncovered in his London laboratory. Fleming didn't invent the relationship between mold and infection. He isolated it, wrote it down, gave it a name science could work with. But the relationship itself had already been observed, informally, for at least 3,000 years before he was born. Here's what's disturbing about that timeline. It means the solution existed the entire time, through plagues that killed a third of Europe, through childbirth fevers that killed young mothers by the millions, through infected battle wounds that killed more soldiers than the battles themselves. The mold that could have stopped a meaningful fraction of it was, in some form, already sitting on loaves of bread in kitchens across dozens of civilizations, just never isolated, concentrated, or understood as medicine, rather than as spoilage to be discarded. A researcher often left out of the Fleming story is French Army Dr. Ernest Duchesne, who in 1897, over 30 years before Fleming's petri dish, documented that penicillium mold could kill E. coli bacteria as part of his doctoral thesis in Lyon. He even proposed it might have therapeutic use in humans. His thesis was submitted, filed, and then largely ignored, partly because he was 23 years old and unknown, and partly because the scientific establishment of the time wasn't looking for what he'd already found. He died young of tuberculosis, a disease antibiotics would eventually be able to treat, just not soon enough to save the man who'd found the mechanism first.
It took two more researchers, Howard Florey and Ernst Chain at Oxford, working through the early years of the Second World War, to turn Fleming's mold into something that could actually be mass-produced. By 1944, penicillin was being manufactured in large enough quantities to treat Allied soldiers on the beaches of Normandy, a drug derived from the same substance that had been sitting on kitchen bread for millennia, discarded as spoilage by everyone who ever threw the loaf away. So here's what the moldy bread actually was. Not folklore, not a placebo people believed in because they had nothing else. An unrefined, inconsistent, genuinely functional antibiotic, sitting in plain sight in kitchens for millennia, discovered independently by cultures with no way to compare notes, because the pattern was strong enough to notice even without a single microscope between any of them available to them. Here's a small etymology aside, worth knowing once. The word antibiotic itself comes from the Greek anti, meaning against, and bios, meaning life, coined in the 1940s to describe exactly this. One living organism waging quiet chemical war against another, except the war didn't start in the 1940s. It started the moment the first mold spore landed on the first piece of bread and began defending its territory against the bacteria trying to take it over. Humans didn't invent that war. They just eventually learned to stand behind the right side of it. 3 tools. Honey that starves bacteria of water and burns them with peroxide. Maggots that eat only what's already dead. Mold that fights bacteria the way it's fought them for millions of years, long before there was a bread or a wound or a human to benefit from it. None of it understood at the time as biology. All of it, somehow, correct. Here's the part worth sitting with after all of that. Every one of these solutions was discovered without understanding. Nobody in ancient Egypt knew what a bacterium was. Nobody packing maggots into a wound in 1800 understood enzymatic debridement. Nobody spreading moldy bread on an infection had ever heard the word antibiotic, and yet the body and the world around it kept producing the right response anyway. Not through knowledge, through pattern, through repetition, through survival doing what survival does, whether or not the mechanism was ever named. What if that's not just true of honey, or maggots, or mold? What if it's true of the responses your own body still runs? The ones that don't come with an instruction manual either. The hypervigilance that won't switch off at 2 a.m., even when nothing in the room is actually a threat. The numbness that shows up exactly when things get too loud, too fast, too much. The overthinking that replays a conversation 11 times before you can fall asleep. None of that was designed by you. None of it was chosen. It was built. The same way the honey's chemistry was built. The same way the maggot's enzymes were built. By something older than your ability to reason about it.
A nervous system shaped by ancestors who needed to notice danger before they understood it, who needed to shut down before they could explain why, who survived first and made sense of it later. If ever, it wasn't weakness, it was mechanism. The same way a wound packed with honey wasn't healing because someone believed in it. It was healing because of measurable, physical, replicable chemistry running underneath a story nobody understood yet.
Here's what's strange about that comparison. We spent 3,000 years treating wounds with something we couldn't explain, simply because it demonstrably worked. And somewhere along the way, we stopped extending that same patience to ourselves. We started expecting to understand why we shut down, why we startle, why we go quiet, before we're willing to accept that we do. As if the body needs a peer-reviewed citation before it's allowed to protect itself. It doesn't. It never did. The bees didn't wait for a Cardiff metropolitan study before their honey started killing bacteria in 3000 BCE. The mold didn't wait for a petri dish in London before it started defending its territory on a piece of bread. The mechanism came first. The explanation, if it ever arrives at all, comes centuries later, or not at all. Ernest Duchesne died without ever knowing that the mold in his thesis would save millions of lives on a beach in Normandy. The mechanism didn't need him to know. It just needed him to notice once and write it down. Maybe that's the smaller, stranger point buried in all of this. Noticing doesn't require understanding. It just requires paying attention long enough to see that something keeps happening the same way every time. So maybe the numbness isn't a malfunction. Maybe the overthinking isn't a personality flaw. Maybe they're closer to what the honey was doing in that sealed clay pot. Old, unexplained, and still quietly doing its job, long after the reason for it was forgotten. Somewhere, right now, there's honey still sealed in a jar from 3,000 years ago. No one's opened it. No one's checked on it. And if they did, it would still be exactly what it always was, unspoiled, still working. Nobody had to teach it how. It doesn't. It never did.